Hi everybody, this is Mr. Nolan, and what I want to show you today is how to do gene expression on paper. We've seen simulators, we've seen, we learn a variety of things about gene expression, but this is really the nuts and bolts. Why does your DNA somehow magically transform into your traits, or how? And so that's what we're going to learn today in this quick screencast. And so this is something that you can follow along with me on the screen. If you have notes or something, you can follow along on some paper. But hopefully this should be able to explain to you just by watching. Our goal is to be able to show the step-by-step mechanism by which DNA codes for proteins in a cell. There's some parts that we have to keep in mind here. Two terms that we need to know is transcription and translation. These are two totally different processes. They're super important, though, for figuring out how to take DNA and to make it into proteins. Your proteins determine your traits. So if you can do this gene expression on paper, what you're really doing is showing here's how DNA results in traits. It's very, very powerful. And it took scientists a long, long time to figure this out. The first thing that you may do is to go to this link that's on the screen. I've also included this in the information section of the video. This is an awesome, awesome film. It's by a team of researchers read by a scientist by the name of Drew Barry. And they created this incredible, incredible video that actually shows this process happening in real time. It's super cool. So if you get a chance, watch it, listen to the narrator. She explains everything that's going on. So watch that, super cool, and then come back here and I'll show you how do we do this on paper. So if you want, take a minute, go click that link, watch the video, and then pause this one. So we want to be able to do this on paper. So there's a few steps. What you might have just seen in the video was that you have DNA in this dark space, the nucleus, and that DNA is read off into mRNA. And then that RNA leaves the nucleus, finds a ribosome, the ribosome reads the codons on the mRNA, and then proteins sort of shoot out. You've got this tRNA involved as well, so there's all these complicated things going on. I want to show you on paper how to do that. No, you don't have to know every single last little bit of those details in order to understand this process. We just have to be able to do this on paper, so I'm going to simulate it by doing it on the screen. There's basically 5 steps. There's things you have to consider in between, but there's basically 5 steps. 1, you have to make mRNA from the DNA. You have to use the DNA to figure out, well, how do I make RNA from this? Then you have to find the start codon on the mRNA. There's a specific codon set of 3 letters that we have to start on every time, and that's because 4 billion years ago when life was first getting off the ground on Earth, some bacterium somewhere said, we're going to start at this codon, doggone it. We're going to start right here, and no matter what, we're always going to start here. And there's really, nobody really knows the reason for this. It's just that there's a certain 3 letters we start at that are very important we need to know. We need to identify where the codons are. That start codon shows us where the rest of the codons are. Super important. That's why we have to identify that start codon. Then we have to use this chart that you see on the right in order to translate the mRNA into amino acids. That's probably the toughest part of this whole process, but even that's not very difficult to do on paper. And then if you hit a stop codon, which you might notice over here, there's these stop codons, that means you're done. You're finished. If you have a pencil in your hand, what you should do at that moment, once you hit stop, is this. Drop your pencil. You're done. If you keep going after stop, if you keep doing more amino acids, you will be wrong. Like, you would answer questions incorrectly if you did that. So we want to stop when you hit a stop codon. So let me show you how to do this. Let me show you how to sort of work through these kinds of problems. Here's an example. We'll do one worked example. Let's say you're handed this stretch of DNA, and it's got A, G, T, C, it's got all these letters. One of the ways that we know that this is DNA is because it has T's in it. T stands for thymine, and T is one of the bases in DNA. There's 4. You got A, G, T, and C. If you find T, that means you're in DNA country. So what I'm going to do right now on the board is I'm going to take the DNA and show you, well, if I were to double this strand of DNA, what would be the complementary letters? DNA is double-stranded. It's a double helix. So if I've got letters on top, what are the letters that go on the bottom? So that's what I'm going to put on the screen right now just to show you what are the complementary bases of DNA. Okay, so I just put up the complementary bases for DNA. So let's look at these for a second. I put up something called Chargaff's rule. This is something that a scientist named Erwin Chargaff discovered in the 50s, that if you have an A on one side of the DNA, you'll have a T on the other. If you have a G, you'll have a C. Right? And then, so that's all you need to know. You can do the rest from there. If you have a T, you'll have an A, and then if you have a C, you'll have a G, right? And so forth. So what I did just now was I went through and I just found what would the other side of that DNA look like. But here's the thing. mRNA is not DNA. It is actually a different, it's a different chemical. It has a different sugar in the backbone, and it also has some different bases. Instead of T's, it's got U's. So if you saw that video, what you would have seen was this yellow strip coming out of the DNA, or out from the DNA. That was mRNA. So in order to make mRNA, it's a little different. It's going to look very similar to the complementary strand of DNA. So AT, G, C, TA, AT, CG. It's going to look very similar, only we replace the T's with U's. So what I'm going to go ahead and do is get rid of this first strip of DNA, because we don't really need to see this right now. This is actually going to distract us a little bit. Okay, so what we're going to do now is we're going to say, we just saw the rules for DNA. Let's apply them to mRNA. mRNA is complementary to the DNA. It's just that instead of T's, it's got U's. So what would that look like? Let's go ahead and put that up. Okay, here's what I did. What I just did was I wrote the letters to the DNA that are complementary, but I replaced the T's with U's. So normally what would go with an A is T. So instead of a T, I replace that with a U. What would normally go with a G is a C. That's normal. What would normally go with a T is an A, that's normal. But again, what would normally go with an A is a T, but down here, mRNA doesn't have thymine. It doesn't have that base. So instead, it's got a U. So if you look really carefully, that's what I did all the way across. I just, and I just drew these lines just so you could tell which letter was talking about what. So you can see that in any situation, A is going to translate down, going to transcribe down to a U. T will transcribe to an A, C will transcribe to a G, and then G will transcribe to a C. So now once we've done this, this is where we have to figure out, well, how do we start this? How do we use this chart over here to choose what amino acids to do? So now that we've got our mRNA, we've just transcribed this. So this is called transcription. We just transcribed the DNA into RNA, okay? What we need to do now is translate it using this chart. So here's how we do it. First, we have to take the mRNA and we have to find the start codon. The start codon, for whatever weird reason, is always AUG. So in the red mRNA, I want you to find from the left, where's the first instance of AUG? Look for it. I bet you found it. It's right here, right? There's our start codon right there. AUG is where we always start. So now that we found our start codon, what we need to do is to identify where the rest of the codons are. Every codon is 3 letters long. So here's our start codon, right there, it's AUG. The next codon would be GAG. The next one would be CUA, and then UCC, and AUG, and UAU, and so on. So what we need to do here is we need to actually identify all those codons. So we're going to split these into 3. So I'm just going to go ahead and underline them. Okay, so each one of these little 3-letter codes that I underlined is called a codon. And each one codes for one amino acid. A protein is made out of a chain of amino acids. So for each one of these sets of 3 letters, I need to choose the amino acid in this chart. I prefer this chart. This is the best chart that I can find to use. The table is a little bit confusing. I really like this one. So to use this chart, all we have to do in order to translate our codons for our mRNA into amino acids is we start with our start, and we take the 3 letters, and we take this chart, and we start from the middle, and we work our way out. So if you look at the start codon, AUG, well, let's look at our codon chart, which is reading the mRNA, AUG. Now, it might be hard to read on your screen, but AUG gives you methionine. And so what we would do for our amino acid down here is we would write methionine. So I'm going to go ahead over to the board, and I'm going to start writing these down. So our amino acid, the first one, AUG, is going to be methionine. Now we can abbreviate that with just a 3-letter term. So we've got methionine. Now let's look at GAG. If I go GAG, we find that we're at glutamic acid. And so we're going to go ahead and put GLU for short. Next one, CUA. CUA, we're going to have leucine, LEU. UCC, UCC, we're going to get serine. Next, we've got AUG, AUG. Look at that, it's methionine again. And then our last one, last couple, UAU, UAU is phenylalanine. We can just do PHY. And then the last one, GGG, GGG, and we get glycine. Sorry, it kind of went off to the side there. So what we had to do was we had to take each one of our codons and we had to use this chart in order to say, well, let's figure out what they actually are. AUG codes for methionine, right? And then we just went through and we went and we found all the amino acids. Now, if you hit a stop codon, which we'll see in our next example, you would immediately be done. You'd be finished. You would not keep going. So this process, turning the mRNA into proteins, is called translation. And so we translated the RNA codons into protein. Okay, so we translated that into that protein. So let's go ahead and do another example. This one will include a stop codon. I'm going to put it on the screen, and I want you to take a piece of paper, and I want you to just try to walk through it. Take the DNA, turn it into mRNA, take the mRNA and use the chart on the screen to try to figure out how do I translate that into the amino acids. Remember, you have to start at AUG. That's the 3 letters you have to start at, and if you hit one of these stop codons, that means you are done. So let's go ahead and move to another screen. I want you to take a few minutes, just pause it, and try to solve this one on your own. And then I'll show you how to do it. All right, there it is. So there's your example number 2. I want you to pause the video right now, and I want you to try to solve this. If you don't have the codon chart, maybe you can see the link below it. It will show you where to go to see a really good example of a codon chart. That's a really good website too. So go ahead and give it a shot. Just go ahead and pause, and then in a moment I'll show you how to go through this example. So go ahead and pause, try to work this one out. Alrighty, so hopefully you have paused it, you tried to work this out in a pencil and paper. I will show you the solution really quickly just to give you a sense of how to do this. First, we have to transcribe our mRNA. Remember, the transcription of the mRNA has to be complementary to the DNA. So let me throw that on up there. What we're going to have down here is that T is going to translate into an A. Over here, the A is going to be a U. We got C, C, G, U, C, U, A, U, G, C, A, U, A, U, U, G, A. So that is our transcribed mRNA. So I'm going to link these up a little just so you can see. All right, this is just so you can see which letters are which. So we just transcribed this. And we have to first find the start amino acid. Shame on you if you forgot to find the start amino acid or the start codon. It's right here. That is our start. So that's our first codon. CAA is our next one. UAU, and then what looks like UAG, or UGA, sorry. So in order to do this, we've got our AUG. That's going to give us AUG, methionine. And then CAA, looks like. So CAA is going to give us glutamine, or GLU. UAU is going to give us tyrosine. And then UGA, look at that. There's a stop codon, so we're going to stop. So what this means is that this is our little protein. Each of those 3 amino acids is part of this protein. And remember, once you hit stop, like I said, you take your pencil and you drop it. So you would not continue translating that. Even if there were more codons here, you would be done. You should not continue if you hit stop like we did over here. So I know it's a little bit of a confusing process. I'm going to send you a link to a website as well that explains to you just what I explained, but maybe you'd find it a little bit more helpful. And also in my information portion at the bottom of the video, below the video, you'll also find links to my website where you can download a worksheet and you can give it a try. So if you were not here today, or if you don't understand this, try to, just figure it out. There's a lot of stuff online. I just gave you a good explanation. So hopefully now you can go through and you can actually do, you can take protein, or a DNA, and actually turn it into proteins, which codes to traits. This is the central dogma of genetics, so we really want to be able to do this.